Tree trunks can support a diversity of organisms. But does a fatter tree trunk support more species than a thinner tree trunk? If we think of tree trunks as being habitats ecologically isolated from each other, kind of like islands separated from each other in the ocean, then island biogeographical theory would suggest yes, there could be a positive correlation between trunk size and species diversity. Island biogeographical theory would also suggest a positive correlation between invertebrate species diversity and bark complexity, as greater bark complexity potentially increases the number and type of niches that more species can capitalize on, like comparing niche opportunities on a flat island with a mountainous one. And could there be an interaction between trunk size and bark complexity in determining species richness? Then there's the small island impact, a theory developed by Lomolino and Weiser in 2001, which states a minimum island size is required before a positive correlation with species diversity can even occur. So, does that mean tree trunks might be limited by a minimum circumference before we see a positive species richness correlation, if we see any at all? It might be that small island impact doesn't apply to all ecological landscapes, as positive correlations have been found for very small areas such as lichen on boulders and in insects in urban roundabouts with higher tree species richness. So, why do we care about this question? Well, it would fill a knowledge gap and expand the application of island biogeographical theory to a new ecological context. But it would also have practical application for conservation of urban invertebrate species diversity. We'd know if we need to protect bigger trees to preserve more species complexity, or if it was just the same to have newer, skinnier ones. To find out the answers to my questions, I did a paired block design examining pairs of Melaleuca quinquenervia, also known as the broad-leaved paperbark. It's a common tree in Sydney with a large range in trunk width and bark morphology. My subject trees could not have major forking branches below 2 metres, they could not have a parasitic plant growing on them, and the canopies of the paired trees could not touch each other. So first I took a lot of photos of paper barks to determine a bark complexity scale from 0, very smooth bark, to 5, very complex bark. Then I travelled around Sydney and measured invertebrate species diversity for a total of 52 trees, made up of 26 pairs of one fatter and one skinnier tree, with each pair separated by at least 1 kilometre from the next pair. I measured their circumference at 150 centimetres above ground level as a proxy for tree size, scored their bark complexity, noted whether they were located on the street verge, in a park or in a reserve. I counted live or dead species, whether they were on the bark surface, hidden within bark cracks or under bark flaps, flew onto the trunk or caught as prey in a web. I also counted egg sacs, cocoons, shed nymph exoskeletons, muddy trails which are evidence of termites, and different types of spider web. I used a probe to gently lift bark and circled each tree 3 times looking for species from ground level to 2 metres high, and wrote them on my data sheets. Then I grouped my data into functional groups: ants, other crawling insects, other flying insects, spiders, millipedes, snails, and then one group of cocoons, eggs, and egg sacs. My hypotheses are that there will be a positive correlation between species richness and trunk size, as well as bark complexity, although I hypothesize that complexity will have a greater influence. I also hypothesize that tree trunks won't need to meet a minimum size threshold to show a positive correlation with species diversity.
My statistical methodology will be plenty. I'm going to perform a paired samples t-test on the 26 pairs of data if data are approximately normally distributed, or a Wilcoxon-signed rank test if data are not, to see if fatter trees have a significantly greater species diversity than thinner trees. But because I didn't have a standard size of fat and thin trees because of the variation in natural tree sizes, I'm also going to perform a linear regression on the 52 individual trees to see if there is a correlation between trunk circumference and species richness, and I'll also do this for bark complexity. Thanks for watching!